Friday. And so a reminder, we have an exam a week from today. We'll have a study session Tuesday, 5:15 in Foster Hall. I'll have to find out which room we can use in Foster Hall. So, and then a reminder, please see the BioCat before or on Friday so you can get that first allotment of 20 points. Okay, so this is what we are also talking about, this REC-A filament, where in the presence of single-stranded DNA, REC-A colonizes it, for lack of a better word, and then is involved in the DNA displacement in terms of getting that. Remember, the context for this in our example is bacteria acquire foreign DNA. And if there is homology with its own chromosome, it can be incorporated into its own chromosome. And so that is REC-A, and a number of other catalysts are the enzymes and catalysts that carry this reaction out, this incorporation of foreign DNA into the host DNA. And so you've got the color-coded, the recipient is in gray, and the foreign DNA is called the donor DNA, is in orange. And we talked about this triplex, where you've got 3 strands of DNA being, one of them displaced and the other one being moved in. And then movement from left to right. So this strand, the REC-A, is looking for homology. And so more and more REC-A piles on and looks for homology. And then the only thing that I thought I would do, and it's not quite on there, is to circle and kind of give you a little bit better feel, or actually look for where this strand lysis is going on and strand ligation is going on. And so I put this little circle. So there is under 5A, this endonuclease activity is cleaving the recipient DNA. And so it is the cut DNA. The DNA that's cut is in gray, which is the recipient DNA. And then the DNA that is being ligated is now the hybrid, the recipient with the donor. And so I said I would highlight that for you last time, and so you can kind of revise your notes there, and even put a circle there where you can see the excision and the ligation going on in steps 5 and 6. And so what I'm not covering, Zach mentioned the holiday structure. After this process of excision and ligation, you now have foreign DNA incorporated into the host DNA. That's the bottom line. So I put this net result in red, donor DNA, and there may or may not be some base differences. The donor DNA may differ somewhat from the recipient DNA, and if so, that recipient host has new DNA, has some mutations in that gene, which may or may not be good. But that is the interesting game that bacteria play in bringing in new genes. Is, oh, maybe this is a slightly modified and slightly better gene that's going to help me grow faster. And then, like I say, the rest of the figure is then the twisting of these 3 strands to resolve itself into 2 strands, double strands each, through the holiday structure to then resolve into a new gene incorporated into the host chromosome. Okay, so any questions on, I'm focusing on REC-A as far as its catalytic ability to invade the strand. It's not the only catalyst, but focus on REC-A and what it is doing in this set of reactions. And we'll talk about it a little bit more in something that Zach mentioned in the SOS response. So the REC-A protein, realize it's multifaceted. It participates as a polymerase enzyme. It's inducing proteolytic activity in the SOS response, and so RECA ends up being sort of the workhorse of this chapter's reactions, because we're going to be looking at it again in a few minutes. Okay, any questions? All right. So, what is a mutation? And I think you know quite a bit about mutations already. But that very first statement is a heritable change in the DNA. You can have mutations happen, and we correct those mutations, and we don't pass them on to our offspring. So, in other words, it has to be a heritable mutation. So, DNA repair, mutations are introduced and repaired. Mutations repaired and are not passed on. And so, the permanent mutation is one that's inheritable. Okay, and that's just to kind of highlight, hey, we repair our DNA all the time, and so mutations that happen can be repaired. So what kind of mutations are there? A point mutation is the traditional either a substitution for a single base pair, and so adenine substituting for thymine, whereas you can have an insertion of a base or a deletion of a base, and they really have much different consequences. You can have inversions where the DNA is flipped in orientation. Reversion, it back mutates to the original sequence. So those are some of the categories of mutations. But another way to look at them is what they cause. What kind of consequences are there? So, for example, if you have a base change in the second base of a triplet codon, that often does not change the amino acid. Or in third base is even more common to not change the amino acid. Because remember, the triplet, the genetic code is redundant, so a number of nucleotide triplets code for the same amino acid. So if you have a mutation and it still gives you arginine, it's a silent mutation. It doesn't change the amino acid sequence. Missense is where there is a change in the amino acid, so instead of arginine, you're now getting tryptophan. You can get a nonsense mutation where the mutation gives you a premature stop codon, and it just stops the protein from being synthesized, and so that's called a nonsense mutation. And then a frame shift mutation is, for example, if you get an insertion of a new base, the entire reading frame is shifted, and so now you're going to get a completely different protein in a frame shift mutation. So the open reading frame is shifted one, and it's going to make a completely new set of amino acids, a new gene that's going to basically have no functional relationship with the original amino acid sequence, the original DNA sequence. So here are diagrams kind of showing you. Here's a missense point mutation where the thymine is replaced with an adenine, and so instead of phenylalanine, you're getting leucine, and so a completely different amino acid. The rest of the protein chain is not changed. And that may cause absolutely no effect. It may benefit that protein, and it may be deleterious. Usually it's deleterious, but it really has those other options. Perhaps that amino acid is not in a functional part of the gene, and it doesn't matter. Okay, so sometimes a nonsense point mutation is where you actually get a stop codon premature, and you just get you stop synthesis. And here's where you get this an insertion, which causes a frame shift. And so you go from AAG, now you insert an AT base, and you get TAT instead of TCC. And because the reading frame is shifted, all the other amino acids downstream are now different. So you get a completely different protein in these frame shift mutations. So you can get a frame shift mutation from inserting a nucleotide or deleting a nucleotide. The reading frame is shifted. And then here's an example of an inversion. And so there's your examples of mutations that happen, and most of these are going to be deleterious, and so DNA repair is so important. Okay. So like I've said, we have our cells, DNA polymerase III, and bacterial cells have the ability to proofread as DNA is synthesized, and has DNA repair mechanisms to correct mutations. Okay, but it's not 100% efficient. Mutations happen, but they happen at a lot lower frequency than they would if we didn't have these DNA repair mechanisms. So then for your information, I'm not really going to cover this, but how do they arise? They arise from these tautomeric shifts in the DNA, deamination reactions, removal of purines. That becomes a little bit biochemical based, and you can cover that in your biochemistry classes. Okay, so mutagenic, either mutagenic chemicals or mutagenic exposure to shortwave radiation. There are things in the environment that causes deamination, causes dimer formation, causes mutations, whether it's chemical or physical. So base analogs, these are nucleotides that mimic the structure of our bases and become incorporated and form a mutation. That's the basis for a lot of drugs that we use, is we use analogs that viruses incorporate into their genome, cause mutations, and allow us to overcome some viral infections. There's chemicals that intercalate the DNA, and that causes, in some cases, mutations. And then even ultraviolet light, short wavelength, and then even shorter wavelength, higher energy ionizing radiation like X-rays and gamma rays either break single-stranded DNA, or it actually can break double-stranded DNA, and then mutations happening. So that's kind of a quick review on environmentally acquired mutations. And I spent a fair amount of time on this next section because Bruce Ames, quite a few years ago, developed a way to screen for mutagenic chemicals to find out whether this red dye number 3 is a mutagen. And if it is, then let's not put it in ketchup, sort of thing. And so the Ames test to identify mutagens is really important. So, kind of the irony of the Ames test is you start with a mutant of Salmonella that lacks one of the enzymes required to synthesize the amino acid histidine. So it is a His minus mutant. And so this mutant cannot grow in the absence of histidine. So start with that. It lacks this enzyme, this mutant, needed to synthesize histidine, so cannot grow in a medium lacking histidine. So then what we do in the Ames test is we have and you're going to do this in the laboratory, is you dip these cotton pads in test chemicals, or you will actually take these cotton pads that are saturated with antibiotics and test the efficacy of antibiotics in the Kirby-Bauer test. And so you'll take, so this is a little cotton disc of paper, and it is dipped in your test chemical. And you put it in the middle of an agar plate. So recognize this as an agar plate with a bunch of bacterial colonies in the middle of the plate and then scattering throughout the plate. So this is your test chemical. And what's interesting is if it is a mutagen, it is going to cause that Salmonella mutant to back mutate and become able to grow on that medium because it is able to synthesize the amino acid histidine. And so that's kind of the ironic thing. So it says the mutagen, so that is the chemical that's in that disc in the middle of the plate, causes a reversion of that mutant back to the wild type, and now reacquires the ability to grow on that agar plate. So this agar plate, you might want to write this in, does not have histidine. And so the mutant can't grow, and the only way that it can grow is if it's exposed to a mutagen. And it actually causes a mutation, and in some of those mutations, it's going to cause a back mutation, and that organism is going to reacquire the ability to grow on histidine plates. And so the positive for a mutagenic chemical is actually the appearance of all of these colonies around this disc. And so that's kind of the ironic result, is the mutagen actually, the mutation allows the organism to grow, and the more it grows, the more potent of a mutagen it is. So we'll take one more look at that plate. So how we do this, and you're going to be doing a similar assay, is you take an agar plate that does not have histidine, and you spread the entire plate with this histidine minus mutant, this organism that cannot grow in the absence of histidine. And then, at the same time, you put the disc right in the middle of the plate. And on that time 0, there's nothing on that plate. So there's your disc, and you've just swabbed this entire plate with 1 billion organisms at time 0. And then you incubate that plate 24 hours. And the agar plate with the disc in the middle. So what happens is the chemical in this disc emanates out by diffusion into the agar, and so you've got this kind of concentration of whatever's in this disc is now in the lawn. And so I'm going to say this is a mutagen. And then we have a plate where you have a control. So you've saturated that disc with water. I'm going to probably end up saying everything here. So that is basically what I'm covering now, but I'm writing it on a board. So you need to use some sort of a control. So water in the disc versus, oh, there's a mutagen in the disc. If this is a mutagen, it is going to cause a back mutation to the Salmonella, and in response to the chemistry right near that disc, these are colonies of organisms that you can now see with your naked eye. And then as the concentration of that mutagen is less, there are less colonies that are, less mutations that are happening further away from the source of mutagens. Whereas in a plate that just has a non-mutagen, you don't get any growth. You don't get any back mutation. And so this is the indicator for the fact that this is a mutagenic chemical. The causing of the growth of this organism. Because in the absence of a mutagen, that His minus Salmonella strain will not grow. And you'll just get an agar plate that's completely clear of colonies with a disc in the middle. But if you start getting growth, it's an indicator for a mutagen. What other was I going to say? Yeah, the only other thing I was going to say is, here's an example of a mutation that benefits the organism. So not all mutations are deleterious. Some mutations benefit the organism. And here's kind of a skewed example of a mutation that benefits an organism. So I think you have all of this information on your outline. So any questions about how the Ames test works? Screening, just simply asking the question, do these chemicals, A, B, C, D, and E, are any of them mutagens? So you do this screen, and it takes like a day to find out. So it's a really marvelous test that Bruce Ames developed and is still used today. So, any questions on the Ames test? It's kind of interesting. The presence of this back mutation means it didn't repair its DNA, because that mutation was heritable. These are all the children, grandchildren, great-great-great-great-grandchildren of the organism that was first placed there. It got mutated, and it passed on that mutation. So it's another example of, DNA repair mechanisms are not 100% efficient. Okay, no questions? We'll move on. One other little variation on the Ames test, to make it a little bit more realistic, is, in the human, we're basically screening for human mutations, human mutagens. And, this is to say, is if it mutates Salmonella DNA, it will mutate our DNA. So we can use Salmonella bacteria as a surrogate. You don't have to do this with people. And of course, that's just one component of our, cancer screening therapy. We use all sorts of test animals. But to make it more realistic for us is when we are exposed, like in our intestinal system, to chemicals, toxic chemicals are detoxified by a huge array of enzymes in our liver. And so to just test this chemical alone and say it's a mutagen is not all that fair of a test. You need, if it's in our food, like if you're testing dyes, that you are food coloring dyes, you actually want to expose it to our liver before you conclude it is mutagenic, because our liver detoxifies a lot of chemicals and changes a lot of chemicals. And so to take that into account, you actually combine that mutagen with a rat liver homogenate, which has this huge battery of enzymes that are going to modify and change, and then you do the same sort of test. So with low, medium, and high doses of a mutagen, if you still get mutations, in other words, if you still get growth of the Salmonella, then you say, okay, even our liver cannot detoxify it, and in our actual human body, even with the liver detoxification system, it still is a mutagen. What's a little bit interesting is sometimes our liver changes a non-mutagen into a mutagen. So that's another reason to do this screen. It's non-mutagenic, except when it's then modified by some of our liver enzymes. It's kind of exceptional. It happens. So it just makes it a little bit more of a realistic, I should say, test rather than text. So, this is something that Zach referred to, and what is most interesting in our DNA repair mechanisms, and now we're really going to switch to bacteria, okay, because we don't typically have an SOS response like bacteria have. We have something similar, but we're talking about what's going on in bacteria now. So we have, our bacteria have error-proof repair pathways, which we have looked at DNA polymerase III, which sees a mistake it makes, which would be a mutation, excises that base and inserts the correct base in their proofreading capability. So that's referring to nucleotide excision repair. There's also a photoreactivation repair pathway that we're not going to talk about. What we are going to talk about is this interesting SOS response, which is prone to a lot of errors, prone to actually introducing mutations, and it really operates only when there is such massive DNA damage that the organism is about to die. So it can't even make a chromosome. So there's times when DNA is destroyed and you're missing 75 bases completely. And you have got to repair that DNA, otherwise you're going to die. And I'm talking about the bacteria are going to die in a matter of minutes. And so huge numbers of mutations are introduced immediately just to repair the DNA. So this is the error-prone pathway, which is the SOS response. And so that's what we're going to be looking at. I've really already covered error-proof pathways, and just in mentioning those, we're going to look at the SOS response, which is an example of the error-prone repair pathway. And so this operates only when the damage is so severe that the cell has no other choice but to die. So you either completely introduce, tens and, I'm going to just say tens of mutations, if not hundreds of mutations. Otherwise, you die. Okay, so you can say, I basically already covered this. So is that a duplicate slide in yours? If it's not, just say, already covered. I'm really not covering any more the error-proof DNA repair. And I want to talk about the error-prone DNA repair. The SOS, save our ship repair. So extensive DNA damage is what induces this response. And again, it's the same RECA protein that in this case has another activity, and that is as a co-protease. And so remember what a protease enzyme does, it digests proteins. And so RECA induces another molecule to be a protease, and so that's why it's called a co-protease. And this activity is induced in the presence of DNA damage. And when you've got DNA damage, it stimulates LEX-A, which is one of these repressor proteins, to actually digest itself, auto-digestion. And so if you have REC-A digesting a repressor protein, that is going to turn on all of those genes that LEX-A repressor is otherwise repressing. Okay, so the digestion of this repressor protein then allows for the expression of a number of DNA repair enzymes. And so there are these sloppy DNA polymerase enzymes that don't do any proofreading. And so we're going to be looking at the UMU-DC gene product, which is one of the polymerases that Zach mentioned, the DNA polymerase 5. And what's interesting about DNA polymerase 5 is it repairs both strands. So if you have actually two strands missing, it can take a ragged end and synthesize two strands. And what is really important is that there's no template there to read the proper message. It's just throwing in nucleotides. And as it throws in nucleotides, it's throwing in mutations. So here is a mutagenic polymerase, which is really interesting. Okay, so here is where these pathways, let's first look at the LEX-A repressor protein. We're going to be looking at how these repressor proteins stop expression of genes in the next chapter. But we've already introduced you to what an operon looks like, and that is a number of genes, or upstream of structural genes is the promoter region. And remember, that is where the RNA polymerase docks onto the promoter region and then synthesizes those genes in that particular promoter. And in this example, this repressor protein is sitting on a number of different operons, preventing their synthesis, preventing their expression of those genes. And so when we have a regulatory protein controlling many operons, we call this a regulon, because there's multiple separate operons controlled by the same regulatory protein. Okay, so there is some background expression of REC-A protein, so a little bit, something like 0.5% of the capable REC-A gene expression is expressed even with REC-A present. And so the real key to understanding this figure, and even to some of Zach's talk, is to understand that single-stranded DNA is the cue for DNA damage. So, of course, DNA is double-stranded. If you have single-stranded DNA, that is the environmental cue, that is the signal for DNA damage. And so if REC-A protein, which we're looking at in this triplet beads of green, encounters single-stranded DNA, that means, hey, there's DNA damage. We shouldn't have single-stranded DNA. Okay, so when DNA damage occurs, single-stranded DNA accumulates, and that's what we mean by activation by DNA damage. Okay, so here is REC-A in contact with single-stranded DNA, and in this form, it causes the LEX-A repressor to auto-catalyze, to auto-breakdown, to actually exert proteolytic activity on itself. Okay, so here is the DNA damage, and it's in this form that the REC-A protein causes the destruction, the proteolytic cleavage of LEX-A. So if LEX-A now is cleaved, all of these operons now are turned on. All of these operons now, all of these, what we call SOS proteins, are synthesized, okay? Including huge quantities of REC-A, for one thing. So the repression of REC-A is now completely off, and you get massive quantities of the recombinase enzyme produced. And the only operon that I'm going to be covering here is UMUDC. It produces the POL5, the polymerase 5 enzyme. And the interesting thing about the polymerase 5 enzyme is that REC-A becomes a component of the polymerase 5. So that is why you see this arrow going from, here's the REC-A, now participates, joins in one of these subunits, and then becomes this UMU-DC product. In other words, becomes DNA polymerase 5. Okay? So DNA polymerase 5 has as one of its subunits REC-A. And what's so interesting about DNA polymerase 5 is, as I've said a couple times, and Zach mentioned it, is that polymerase 5, number one, has no proofreading capability, so it makes mistakes. But the main thing it does is it's able to synthesize de novo 2 new strands of DNA. And by definition, if you are not using a template to see what bases should be incorporated, every base you incorporate is a mutation. So error prone is stating it mildly. It's nothing but errors in terms of introducing mutations. But bottom line, it's repairing a broken chromosome. And if you don't just make those connections again between, if you're missing 50 bases completely on both strands of DNA, you are not going to be able to perform replication, and that cell is going to die. And so that's why it's called the SOS response, because sure, I'll take 50 or 75 mutations in that one gene, or a couple of my genes might become mutated as a result of this DNA polymerase 5 error-prone activity, but I live another day. And so that's why it's mutate or die. So it's a really interesting example of the quote, purposeful introduction of huge numbers of mutations in the context of, well, it's either that or I've got about 5 minutes to live. And so if I can repair my chromosome, I can copy it, and I can make 2 babies. Well, they've got massive mutations, but, well, that's their problem. No, they don't say stuff like that, because they don't say anything, right? Okay, let me see. Yeah, I'm going to talk about horizontal gene transfer. So understand, the main thing I'm focusing on as far as the operons here is the UMU DC gene, which makes this amazing enzyme called polymerase 5, and that REC-A is a subunit of that DNA polymerase 5, and that is the enzyme that introduces mutation. Okie doke. I've got your exams that people didn't pick up last time, and so people who need to do that, come and get